Good afternoon, friends. Myself Rana Archana. I am assistant professor in Sri M.N. Virani Science College, Rajkot, and my colleague today, Neha Vanjara. She is from Christ College, Department of Microbiology. We both are going to conduct a short lecture on the topic biofertilizer. So today we will discuss with you biofertilizer, one of the very current topic, biofertilizer. So we are starting our discussion. So first of all, before we proceed for the discussion of biofertilizer, we have to discuss about the plant. So you can see from the slide what exactly the slide shows. The plant growth that carry out the reaction photosynthesis, that is CO2 plus H2O, that give rise C6H12O6. The carbohydrate synthesis is going to be there. The sunlight is required, the radiant energy utilizes, as well as the soil is providing the substratum. And we know very well the water is required, and water is provided by the rain, as well as that is provided in the various form of the precipitation. So whatever is there, that is not only the thing that we are observing. The soil is not providing only the substratum. You can see in the slide in the bottom N, P, K, C, A, M, G and S, that is shown in the slide. What exactly the N? Nitrogen, phosphorus, potassium, calcium, magnesium, sulfur, and also there are many others. Micronutrients are there. They are also required for the plant growth. So soil is not only the substratum, soil is also providing with its many type of the macro and micro elements that is required for the plant growth. So these nutrients are taken up by the plants, that is taken up from the root, and by that the growth of the plant is there. So our today's topic is biofertilizer. So we have to first of all discuss about the soil. You can see in this slide also the soil is there. Because of the soil, not only the growth of plant is there, but in the soil crop residues are there, sewage, sludge, feed or food processing waste is there, the manures are there. They are going to be mixed inside the soil, and because of that whatever the reached soil is going to form, that is providing the nutrition. So soil is the one that provides the nutrition to the plant. We are thinking that the soil have all the elements in them, but once when we are proceeding for the crop, at that time that may be utilized by the plant, and that's why the fertilizers are important. So today we are going to discuss about the fertilizer. So first of all, fertilizer. What exactly the fertilizer is? A fertilizer or manure are material that contain one or more of the essential element and are applied to the soils to encourage the crop growth. It means fertilizers are required for the crop growth. Without fertilizer, the crop may be not going to grow in such a way they are growing. That's why for the growth fertilizer is significant. Most of the fertilizers sold contain nitrogen, phosphorus and potassium, either singly or in the combination. What it means? Nitrogen, phosphorus and potassium are required element for the plant growth that may be applied in the single form, maybe in the combination can be applied via fertilizer. Now one question may be arise in the mind, what exactly the role of fertilizer? Why to use the fertilizer? The simple answer is there, to enhance or to increase the productivity. So for the productivity enhancements, farmers has to use the different types of the fertilizers, and that's why the fertilizers are there. They are there in the market, that is available in variety of the form, the chemical fertilizer, organic fertilizers, inorganic fertilizers, and with that one important is that biofertilizer. So use of the balanced fertilizers should be there. Use of balanced fertilizer can provide viable leverage to increase crop productivity. It enriches the soil with important nutrients. That's why the biofertilizers are significant. So what exactly the thing is, balanced fertilizer should be used. It means we can go for the use of organic, we can go for the use of inorganic biofertilizer, we can even proceed for the use of the soluble as well as insoluble type of the biofertilizers. But important and current aspect is use of biofertilizer is significant, and that's why we have to come to the point how they are significant as compared to chemical fertilizer. In the market even chemical fertilizers are also available. So chemical fertilizers are there, they are giving the more crop production within the short time, but still we have to use the biofertilizer, because in comparison to the chemical fertilizer, they are eco-friendly. They are not producing any pollutant that is produced by chemical fertilizers. So first point we have to keep in the mind that chemical fertilizers are such, they are producing different type of the pollutant that is not going to be produced by the biofertilizers, as well as they are, by production point of view, they are cheap. They can be produced by easy way, so production is also easier, and that's why we have to proceed for the discussion of biofertilizer. You can see from the slide that biofertilizer, it means what? Bio, it means life. So live fertilizer, jene jaivik khatar ke chhe. So live fertilizer is the one that is applied and the life is going to form the soil fertile, and that's why that is known as biofertilizer, the live. So a biofertilizer is a substance which contain living microorganisms. Living microorganism may include bacteria, viruses, fungi, algae, as well as different type of the protozoa. How they are applied? They can be applied to the seed, they can be applied to the plant surface, they can be applied to the soil also. So their application can be there by either of any of these three modes. But what they are doing, they are enhancing the crop. They are enhancing the particular income of the farmer. So that has been shown in this particular slide. You can see from the picture, this particular farm is there, that have the high crop. That is not only because of fertilizer, but biofertilizer is added in that particular one. So farmers' income can be enhanced by the use of biofertilizer. So it inputs containing efficient strain of specific microorganism which are capable of mobilizing nutritive elements required for plant by fixing the atmospheric nitrogen, by solubilizing and enhancing take up of the soil phosphorus, and by stimulating the plant growth through the synthesis of promoting or growth promoting substances. So what thing is written in the slide, you can just see. Biofertilizers are significant because in that the microorganisms are present which can fix nitrogen, which can solubilize and enhance the take up of the soil phosphorus, as well as the stimulate the plant growth, and that's why the use of the biofertilizer is there. Now we have to come for the discussion of types of the biofertilizer. This is very important topic because we know the bio, it means life. So how we can apply the life for the enhancement of the crop, and that's why there are many type of the biofertilizers available in the market, and we have to discuss about that particular one. You can see from the slide. The list is there of the types of the biofertilizer. The first one is nitrogen fixing bacteria, then phosphorus bacteria, mycorrhizae, compost, biocompost, Vermi Compost, Trichoderma, blue-green algae, as well as still many more type of the biofertilizers are there. And now we have to proceed for the discussion of this each and every individual one with their details, how they are proceeding as a fertilizer. So first one we are coming to the discussion, nitrogen fixing bacteria. Nitrogen fixing bacteria are basically of two types, the one known as rhizobium and the other that is known as azotobacter. Rhizobium are not free living bacteria like azotobacter. Azotobacter fixes nitrogen, they are free living bacteria present in the soil, while rhizobium are the bacteria, they are remain attached with the leguminous plant. You can see from the diagram also, in the diagram in the picture, the roots has been shown that have the nodules in them. These nodules are having in them the structure of the bacteria that is known as bacteroid, and which have the nitrogenous enzyme complex which is responsible for the nitrogen fixation. So nitrogen fixing bacteria are of two types, rhizobium that is symbiotic association with the plant, and other azotobacter, they are free living. So if we apply these two types of the bacteria to the soil or to the plant, then they will fix the nitrogen which is not available in sufficient amount that is required by plant, and by enhancement of the nitrogen requirement the plant can be grown. So application of the rhizobium and azotobacter is applicable and is significant for the plant growth when nitrogen is required by the plant. So generally leguminous plants require more amount of the nitrogen, and at that time this should be applied. Now the rhizobium and azotobacter, whatever are the bacteria, just like that, the next type of the bacteria is there, that is known as phosphate solubilizing bacteria. The solubilizing unavailable organic or inorganic form of phosphorus that may be up to 80%. So PSB, they are known as PSB, phosphorus solubilizing bacteria, that may be bacillus, may be pseudomonas, or may be acetobacter. So they can be isolated from the soil also, and they can be applied as a biofertilizer. You can see in the picture the Petri plate has the growth of the organism, and zone of clearance is there, that is also known as zone of utilization. The organism has utilized the particular area that has been shown in the plate where phosphorus utilization is there. So that may be organic or inorganic form of the phosphorus is there. The utilization aspect may include the formation or production of organic acids, by which also this type of the zone of utilization can be there. Organic phosphorus slowly mineralized by the phosphatases. So which enzyme is responsible for phosphorus solubilization? The name is phosphatases. And inorganic phosphorus solubilized by action of organic and inorganic acid. And this is the point of discussion, that organic and inorganic acids are such which can solubilize the unavailable insoluble form of the phosphorus that can be utilized then by the plants. So many bacteria like bacillus, pseudomonas can be applied as PSB, phosphorus solubilizing bacteria, which are responsible for phosphate solubilization. It is not necessary that a particular phosphorus biofertilizer is used for a particular type of crop. They can be applied for any type of the crop. So they can be applied for any crop. Now the third type of the biofertilizer, very significant and widely available in the market, that is known as mycorrhizae. Biofertilizer and mycorrhizae. What exactly the mycorrhizae? The term mycorrhiza was taken from the Greek language meaning fungus root. This term was first coined by Frank in the 1885. Fungus root. Why the name is fungus root? Because mycorrhiza. Myco it means fungi, rhiza it means root structure. This fungi is found in the association with the plant root, and that's why they are known as mycorrhiza. The mycorrhiza is mutualistic association between fungal mycelia and the plant root, and that's why they are known as mycorrhiza. Biofertilizers and mycorrhiza are very important to any revegetation effort, as they help to rebuild the living soil that can get damaged by the earthwork. So what exactly the thing is? Revegetation, it means what? Many of the time soil is, in the previous time it was fertile, but because of the typical type of the work, earth is damaged in such a way that cannot able to grow the plant. At that time, if mycorrhiza is applied as a biofertilizer, then revegetation can be possible. Revegetation, it means again you can proceed for the different type of the plant cultivation. And at that time, mycorrhiza is very efficient strain which is able to grow such a crop that cannot be cultivable after the different type of the damage that has been made up to the soil. So biofertilizer and mycorrhiza is very significant association, and that should be there when any damaged soil is there. Now we are coming to the discussion again. That particular mode of action is what? They are maximizing the plant absorption area and they improve the soil texture. What exactly the meaning of plant absorption area? Plant absorption area, it means the plant is taking the nutrient from the roots only. Now root is present in the soil substratum and it is responsible for taking up of the nutrient. But root is not such a much more wide area. At that time, mycorrhizae is coming into the association with the plant root, that is enhancing their surface area in such a way the more and more nutrient is going to be taken up by the mycorrhiza and the plant is going to grow more and more. So maximizing the plant absorption area, even improve the soil texture, even increase the water uptake in the plant, even increase the mineral uptake, and because of that they are most widely useful biofertilizer because they are giving phosphorus, copper as well as zinc type of the mineral that is unavailable to the plant otherwise. Now there are two types of the mycorrhiza, Ectomycorrhizae and Endomycorrhizae. As the name suggests, ecto and endo. The ecto, it means outside, and endo, it means inside. So now we have to discuss this two type of the mycorrhiza. The Ectomycorrhizae that form a sheath around the root. It means they are making just like the coating of the root. They are not penetrating inside. The mass of hyphae is equal to mass of root. You can even see from the picture. This particular figure or picture shows the particular growth of the mycorrhizae on the root surface. Hyphae grow between the space of the cortical root cell only. They are not penetrating inside, and that's why they are ectomycorrhizae, not penetrating inside. No cellular penetration by the hyphae. Reproductive spores are formed in soil only. They are not produced inside the plant. They are forming on the soil only. And members of basidiomycetes and ascomycetes generally act as a ectomycorrhizae. Just like that, another is there, that is known as endomycorrhizae. The hyphae form arbuscles, that is bulb structure, where they penetrate inside the plant cell. They are also known as VAM, vesicular arbuscular mycorrhizae. What is VAM or AM fungi? First of all, endomycorrhizae is also very well known as AM fungi or VAM fungi. What is VAM and AM fungi? Endomycorrhizae, they are penetrating inside the root cell. They are taking up the nutrient from the soil, applying to the plant each and every cell, and by they are enhancing the crop production. Direct cell to cell exchange of the nutrient is there. Hyphae grow into the intracellular and intercellular space of the cortical cells, and this is the schematic diagram of the vesicular arbuscular mycorrhizae. You can see the different squares are there, that is of the plant cell representation, and the particular line is there that shows the mycorrhizae penetration inside the plant cell. They are penetrated inside in such a way, they are giving each and every type of the nutrient that is taken up by them to the plant cell that is otherwise unavailable to the plant. So vesicles is formed inside, and that's why that is known as vesicular arbuscular mycorrhizae. So that is endomycorrhizae. They can form spore inside as well as outside of the plant cell. The same point we have discussed in ectomycorrhizae, that they cannot form spore out inside. So here at the both side they can form the spore. They can infect grasses, most herbaceous dicots, many shrubs, as well as they can infect to the different genera of the gymnosperms. This is the same thing but with the different presentation, mechanism of establishment of endomycorrhizal symbiosis. You can see how they are penetrating inside and how they are leaching the particular nutrients from the soil, and how they are giving in the form of vesicles to the plant root, and by the plant root that is supplemented to the each and every cell of the plants. Now we are coming to the next type of the biofertilizer after the discussion of nitrogen fixer, after the discussion of phosphate solubilizer, after the discussion of mycorrhizae. The next type is compost biofertilizer. The word compost is very well known. Compost biofertilizer are those which make use of animal dung to enrich the soil with useful microorganisms and nutrients, and this technique is very well known in many of the villages. How compost happens? You can see in the picture the air, water, different earthworms, insects from the microorganism, dead materials, litters, bacteria, fungi and other microbes, even the leaves, even the woody material, even the different food scraps, even the grasses, all combine, and finally what they make? Compost. So finally making up of the compost is going to be there in the particular routine day-to-day life also, and this compost can be utilized to enhance the plant growth, and that's why compost is one of the significant biofertilizer. So to convert the animal waste into the biofertilizer, the microorganisms undergo the biological processes and help in breaking down the waste. And this is the same picture that shows the waste is collected, and when you are applying microorganisms in them, the compost can be formed. The cellulolytic fungal culture and azotobacter culture can be used as a compost biofertilizer. This type of the compost are available in the market, which can be applied for the enhancement of the plant or the crop production. Just like that, the next type of the biofertilizer is there, that is known as Biocompost. It is a kind of organic fertilizer which is prepared from the waste of sugar industry. Compost is the one which is animal waste, and this is the waste of sugar industry. The waste is decomposed using a number of human and plant friendly bacteria and fungi, and this even can be applied to the crop enhancement. Biocompost consists of nitrogen, phosphate solubilizing bacteria and plenty of useful fungi like decomposing fungi. So what exactly the thing with the combination to the nitrogen fixing bacteria with the combination with phosphate solubilizing bacteria, the biocompost can be applied for the enhancement of the plants. This biofertilizer helps the farmer to increase the soil fertility and thereby increase in the yield of crops. The next is Vermi Compost. Vermi Compost is stable, fine, granular organic matter. When added to the soil, loosens the soil and provides the passage of the air. You can see in the picture different type of the worms are there. The mucus that is attached to the cast being hygroscopic in the nature, which improve the WHC, that is water holding capacity of the soil. આપણને ખબર જ છે that we know very well અળસિયું એક જે કોઈ પણ ખેતર છે જે ખેડે છે એ ખેડૂતના મિત્ર તરીકે ઓળખાય છે. And that is the same thing that is there, that particular concept is utilized in the preparation of vermi compost. By the application of different worms, the soil is going to be the fertile in such a way that have the high water holding capacity as well as many types of the nutrient that otherwise does not available to the plant as such, and that can be applied by vermi compost. So it is also an organic fertilizer containing nitrogen, phosphorus, potassium, sulfur and organic carbon, etc. You can see over here also many type of the worms are there. So vermi compost can be prepared which have the nitrogen, phosphorus, potassium, sulfur as well as different type of the carbon that is required for the plant growth. If used over a period of time, the soil becomes extremely fertile and all the lost nutrients are restored back and soil remains fertile. So vermi compost is the one, if you use for the particular period of the time, then soil fertility can be maintained and whatever the nutrient has been taken up, that has been leached out from the soil, that can be again restored. So that is also beneficial. That is the one, that is vermi compost. The next is Trichoderma. It is an eco-friendly fertilizer which acts as a biocontrol agent and is hyperparasitic, different pathogen in the field. So there are many types of the pathogen that has been found in the different type of the soil that can be overcome by the help of Trichoderma. Many of the time what is the thing, biofertilizers can be added with the pesticides also, fungicides also. So Trichoderma is also assisting by that particular way. There are many other biofertilizers are there. Industry has launched many new biofertilizers. They have been found to be effective and needs to be promoted like blue-green algae, Acetobacter diazotrophicus, that is endophytic nitrogen fixer in sugarcane industry. The next is zinc solubilizer, that is the Pseudomonas, Bacillus and Thiobacillus species. Potassium mobilizer, that is Bacillus species. So production of potassium fertilizer can be there by application of the Bacillus. Even production of the zinc solubilizer like Pseudomonas, Bacillus, Thiobacillus can be applied for the production of the biofertilizer. Even the blue-green algae can be applied as a biofertilizer and Acetobacter diazotrophicus can be applied as a biofertilizer. Even effective microorganisms like yeast, lactobacillus and rhodopseudomonas in the combination are excellent for quick residue degradation and fixed nutrient solubilization. So what exactly the thing is? There are many nutrients available in the soil. Whatever the nutrients are available in the soil, maybe plant are not able to take up that nutrient. So these microorganisms are assisting in such a way they become available to them by solubilization, by their action, by endo or by ecto action, it means by inside action or by outside action, and because of that they are making available that nutrients for the plant growth, and because of that crop production can be enhanced. So biofertilizers are such that we have discussed that all are the types of the biofertilizer. So up to this whatever we have discussed, that is the introduction of biofertilizer, what exactly the biofertilizers are, and different types of the biofertilizer in that particular one: nitrogen fixing bacteria, phosphorus solubilizing bacteria, then mycorrhiza, endomycorrhiza, ectomycorrhiza. Then even we have discussed about the biocompost, compost, vermi compost, trichoderma, other different type of the biofertilizers. All this type of the biofertilizers are significant, but how they can be produced? How they can be produced at the large scale? So production technology and method of applying this biofertilizer, that is going to be discussed by my colleague, that is Neha Vanjara. So now the further whatever the talk is there, that is going to be conducted by Neha Vanjara. So please.
Hello friends. Now my friend Archana, she has given basic idea about biofertilizer, types of biofertilizer. Now I will start my discussion with how can we produce them, types of biofertilizer on the basis of their production technology. Now whenever you will refer slides, on these slides you can get idea that whenever we'll see production technology, that whenever we are going to produce biofertilizer, in which form we can produce. So whenever we are going for production, we can produce biofertilizer in two forms, that is carrier based biofertilizer and liquid based biofertilizer. Now what is carrier based biofertilizer? Now generally carrier based biofertilizers, they are solid in nature because they are prepared with the help of some solid supportive material, that is here we may use activated charcoal, which will act as a carrier which will continuously release, and it will allow slow release of microorganisms. Now next one, that is liquid based biofertilizer. Now it is, as such name suggests, it is liquid by formulation and it contains dormant form. So whenever we'll see carrier based biofertilizer, they are solid in nature, but this is liquid. Carrier fertilizers contains vegetative cells of microorganism, but these liquid biofertilizers are having dormant form of desired microorganisms and their nutrients. So whenever we'll input them in the soil, what they will do? Because whenever we are going to apply, they are there in their resting form, that is spores and cyst. So in that form they will remain available for a longer period of time, so their shelf life is very high, and in that form they can able to remain survive for a longer period of time. Next. Now whenever we'll apply them in the soil, what they will do? By the process of germination, activation and outgrowth, they will be converted to active cells, and these cells will grow and multiply by utilizing nutrients which are remain present in the soil, and by that they will remain beneficial for the plant rootlets. Now whenever we want to decide or whenever we want to produce any type of biofertilizer, we have to check two parameters, because efficiency of biofertilizer, it is dependent on two things. First one, number of cells, because if number of cells will not be enough in number, then definitely our crop production will not increase up to the mark. So first thing, number of cells should be optimum, which will improve production capacity of the plants. Second one, efficiency of the microorganisms to do their work. If we are going to use nitrogen fixers, then it must be able to fix nitrogen in proper amount. Same way, if we are going to use phosphate solubilizers, then they should be able to solubilize phosphate. So after addition in the soil, they must remain active, then only our goal gets satisfied. Now, so whenever we are trying to understand advantages of liquid biofertilizers, there are so many advantages. Now, as we have discussed earlier, liquid biofertilizers are having dormant form of cell, that is spore or cyst. That's why definitely their shelf life will be long, that is up to 12 to 24 months. Now, as they are dormant, they are quite resistant bodies. That's why there will be no effect of high temperature, or chances of contamination will be less, because we can store them up to the temperature 450 degree. So at that temperature, definitely normal organism like mesophilic or psychrophilic, thermoduric, they can't grow. Only thermophilic organism can able to survive, and not all thermophilic, only hyperthermophilic organism can able to survive at that temperature. That's why definitely chances of contamination will be less. Now it is easy to use by the farmers. Now beside this, we can able to maintain high population, that is 109 cells per ml we can able to develop. Now how it is possible? Because whenever we are going to develop liquid biofertilizer, they are having dormant spores. Whenever they will get nutrients which generally remain present in the soil, they will start activation, germination and outgrowth, and by that they will be continuously converting to vegetative cell, which is the active form of cell. That's why we can able to maintain high density of cell. Same way as they are dormant form, we can easily export such type of biofertilizers, and here as they are highly active, we can reduce the dose, means here doses are 10 times less than carrier based biofertilizer, and here whenever we'll see quality control protocols are very easy as well as very quick. So now in that case we have discussed advantages of liquid biofertilizers. Now whenever we'll try to refer disadvantages of both these types of biofertilizer, now first we'll see which are the disadvantages of carrier based biofertilizer. Generally as they contain only vegetative cell, shelf life will be low. Same way some vegetative cell, mainly if we are going to use thermophilic, then only they are temperature resistant, otherwise all the other organisms are temperature sensitive. Here if we are going for normal growth pattern or normal environmental condition, definitely chances of contamination will be high as compared to liquid based biofertilizers. Same way whenever we are trying to refer disadvantages given by liquid biofertilizer, as such they are maximally advantageous, but here main problem, that is high cost, and whenever we want to develop such type of biofertilizer, we need high investment for the production unit. Now whenever we'll compare, advantages are more with liquid biofertilizer. So liquid biofertilizers, these are the solution on carrier based biofertilizers. Now whenever concept of biofertilizer get evolved, at that time mainly research was going on to produce carrier based biofertilizer, but now we are having idea that liquid based biofertilizers, they are more important, they are more useful, they are more advantageous as compared to carrier based biofertilizers. So nowadays research are going on to start production and testing of liquid based biofertilizers. Now whenever we want to produce any such type of biofertilizer, we need some equipment. So now we are trying to discuss about equipments. Now first and most important equipment which we need for any of the microbiology practical, that is autoclave, because in microbiology as microorganisms remain present everywhere, first of all we have to allow sterilization of each and every object which we are going to use for microbiology practicals. So here autoclave, it is the apparatus which will allow sterilization of the necessary things which we have to use for our practical, that is mainly nutritive media, all the glasswares and all. Now here whenever we'll see principle behind the functioning of autoclave, it will mainly work on steam under pressure, because as you know that boiling point of water, that is 100 degree. So whenever we'll just boil water, it will give temperature up to 100 degree, but if we'll try to produce steam, and this steam whenever it get packed in specific vessel, it will generate pressure. So autoclave is an apparatus in which we can increase pressure, and by that we can able to increase temperature. So steam under pressure is the main principle which will work behind autoclave. And here whenever we want to remove vegetative cells from all the necessary things which we are using, we have to autoclave them first. Now another instrument which we can use, that is laminar airflow chamber. Now here what happened, laminar airflow, once we'll autoclave all these necessary things, after that we have to start our work, that is we have to inoculate specific group of organism in nutritive media. Now here we have to use such type of instrument in which now contamination can't get enter. So laminar airflow is having one specific filter, that is HEPA filter, so it will continuously pass sterile air. So now chances of airborne contamination get decreased, and that's why we have to use this instrument. Now next one is BOD incubator. Now whenever we want to allow controlled growth of specific group of microorganism, at that time we have to incubate them in specific incubator which will provide specific amount of light, temperature and humidity, and it will increase multiplication of our starter culture. So within a short period of time we'll get very high growth of microorganism. Next one is fermenter. Now fermenter is the equipment which provides proper environment for a growth of desired microorganism. It is generally a large vessel in which organism may be kept at required dissolved oxygen, temperature, pH, concentration and substrate concentration. Now whenever we'll try to refer types of fermenter, mainly two types of fermenter we can say these are available. Simple fermenter, in which it contains steam generator which will try to decrease chances of contamination, sterilization process device, and agitator which will continuously mix the content, and another one that is sophisticated fermenter which contains different biosensors. These biosensors are pH regulator, oxygen level regulator, antifoam device, temperature controller, etc. Now beside these equipments we have to use some other equipments also, that is rotatory shaker which will continuously giving shaking condition and by satisfy oxygen need of the organism. Hot air oven which is generally used to sterilize glasswares like pipettes and all, because it will work on dry heat. So dry heat is the main principal agent which will allow sterilization of glasswares. Then pH meter, because whenever we want to allow growth of organism we have to give them proper pH. That's why before using nutritive media we have to check pH of each and everything. And then refrigerator, after getting proper amount of starter culture then we have to reuse them. That's why we have to store them properly. So for preservation mainly we have to use refrigerator, because of low temperature it can able to maintain our culture for a longer period of time. So now by that we have discussed all the equipments which are necessary for the production of biofertilizer. Now whenever we want to go for mass production, now here time will not permit us to discuss in detail processes of mass production of biofertilizer, but which are the stages which we have to cover, that will discuss only. So here mainly we have to cross 3 stages, by that we can able to go for mass production of biofertilizer. First one is culturing of microorganism. The microorganism which we want to use, that we have to culture. For example, if we are going to use nitrogen fixers, then we have to use either rhizobium or azotobacter. So in that case first of all we have to allow culturing of this organism. Then processing of carrier material. If we are going to produce carrier based biofertilizer, then we have to select carrier, because it should be functional properly, then only it will help our organisms to work potentially. That's why then we have to choose carrier molecules. Third thing, that we have to mix them properly, that mixing the carrier and the broth culture, and after that we have to package them under sterile condition. Now so by that we have discussed production, how can we produce, how many different types we can produce, how many equipments we need. But now we have to check the methods, because after getting production or after getting types of biofertilizer we have to apply them in the field. So which are the different methods available by which we can use them. So now we are going to check methods of application of biofertilizer. So now whenever we'll see methods, there are mainly 4 methods which are available. Now here whenever we are going to use methods, we can apply biofertilizer either on seed or on plant surface or on soil. So here which are the methods? Now it is the list of methods of biofertilizer. Now here mainly 4 methods are there in the application of biofertilizer. But whenever we are going to use any of the method, we have to keep some points in our mind, that this method should be practical, effective and cost efficient. Same way method used affects nutrient availability of plant use, and fertilizer must be dissolved, then and then only it will act properly and it will enhance plant growth. So now which are the methods available? There are mainly 4 methods: seed inoculation, root and seedling treatment, soil application, and then self inoculation. Now we'll start our discussion with seed inoculation. So now here name itself suggests we have to allow application of biofertilizer on seed. So here we have to mix 10% solution of jaggery with biofertilizer. This slurry is then poured over the seeds. So now after that, after mixing them, we have to spread them on cemented floor and mixed properly in a way that thin layer of jaggery will form around the seeds. Now after that we have to allow it to dry in the shade overnight, and then they should be used. Now whenever we are going for this method, generally 750 gram of biofertilizer is required to treat the legume seeds for 1 hectare area, and this is widely used, most common method of the application of biofertilizer. Now next method, that is root and seedling treatment. Now here for all the plants we are not going to use seed. Now in some cases we have to directly use root and root seedlings. So in that case we have to go for this application, that the seedling roots or of transplanted crops are treated for half an hour in the solution of biofertilizers before transplanting in the field. So in this method, seedlings required for 1 acre are inoculated using 2 to 2.5 kilogram of biofertilizers. For this, in a bucket having adequate quantity of wet water is taken and biofertilizer is mixed properly. So first of all we have to prepare proper solution of biofertilizer, and then roots of the seedling are then dipped in this mixture so as to enable roots to get inoculum. And whenever we are going for plantation, now it is rich in terms of biofertilizer and it will give us best result. Now then we have to discuss that what they will do. So these seedlings are then transplanted. This method has been found very much suitable for certain crops like tomato, rice, onion, cole crops and flowers. Next method is soil application. Now sometimes we are not going to use seed treatment or root and seedling treatment. Instead of that we are adding biofertilizer in the soil. So this method is mostly used for fruit crops, sugarcane and other crops where localized application is needed. At the time of planting of fruit trees, 20 gram of biofertilizer mixed with the compost is to be added in the ring of one sapling, that is rootlets. Now in this method what we have to do, that here directly we are mixing it with the soil. So here also first of all we have to prepare solution. That's why here we have to take water in the drum, sufficient amount of water we have to take in drum, and then we have to mix our biofertilizer, and that we have to use in the application of soil. Now here in this case we can modify this technique by broadcasting. Now broadcasting means what? Here we have to use spreading method. So before broadcasting, the inoculum should be incubated with the desired amount of well decomposed granulated farmyard manure. Now what is farmyard manure? Now whenever we'll see manure is an organic material which will promote plant growth. Now manures are of different types. It may be plant origin or animal origin. So farmyard manure is a type of animal manure. So here what we have to do, we have to prepare one plant bed in which we have to try to absorb animal feces, animal urine, and by that we can able to prepare FIM. Now here FIM act as a food as well as adjuvant. So adjuvant means carrier which will allow slow release of organism, and because of that it will remain active for a longer period of time. Okay, so now here whenever we'll see broadcasting, it is just spreading, and here figure is already given which will show you that by that we can able to cover entire production area. Now next one is also the figure by which we can able to cover entire area, and by that we can able to improve production capacity of the plant. Here also some figures are there by which we can able to apply biofertilizer by different methods. Now next one is self inoculation, which is last technique by which we can able to apply biofertilizer. Now this method is exclusively suitable for the application of azotobacter. Now in this method 50 liters of water is taken in a drum and 4 to 5 kilogram of azotobacter biofertilizer is added and mixed properly. So it is widely used for azotobacter species. Now here what happened, in some plants we are not going to use seed or plant rootlets. Instead of that we are using some other things. For example, if we are going to treat potatoes, then we have to use tuber, that we have to dip in this fertilizer, and then we have to allow it to dry. Then we have to allow it to dry in for shading for overnight. We have to incubate them, and finally we'll get material which is ready for plantation. So now this is the method. Now here these many methods we have discussed. So mainly 4 methods we have discussed: seed application, root and rootlet or seedling application, self application and soil application. But which are the corrective measures needed in application? Now sometime what happen, if we are going to use just one type of method, we can't get maximum benefit. So here there are some proposals are given by which we can able to improve crop production. So now either we can use both seed treatment and soil treatment together. Sometimes we can use soil treatment by mixing with compost. Sometimes whenever we are going to use phosphate solubilizing biofertilizer, we have to use it with soil treatment, same way with compost. So now here what is the case? Instead of going for single treatment, if we are using mixed application of biofertilizer, it will be more beneficial and it will promote crop production as compared to all the other things by higher rate. Now these are some figures of fertilizers in which here packaging, transportation, production and their application, all these things are mentioned. Now here we have discussed enough things about biofertilizer, that Archana has given idea that what is biofertilizer, types of biofertilizer on the basis of their function, that whether they will fix atmospheric nitrogen, whether they will allow solubilization of phosphorus. Same way I have started my topic with the discussion of types of biofertilizer on the basis of production technology. But after all these things, finally this biofertilizer should be used by farmers. So now next point is, how can we encourage farmers? So now if we want to encourage farmers, first of all we have to make easy availability of the biofertilizer, because nowadays what is the case, majority of biofertilizer is being routed through government agencies, which needs to be discontinued because sometimes hurdles are created to reach to the farmers. So here we have to start encourage procedures to develop direct marketing channel and supply to farmers directly. Now subsidy can be provided on the lines of chemical fertilizers to the manufacturers, to the end farmers directly. Now which are the advantages? So here already we have mentioned that it will fix atmospheric nitrogen, increase availability and uptake of nutrients, stimulate plant growth, they are cheap, hence reduced cost of cultivation, improve soil properties, lead to soil enrichment, compatible with long-term sustainability, build up soil fertility. Now all these things we have already discussed, but whenever we are trying to refer disadvantages, now as such there are no disadvantages of biofertilizer, but what happens if it is not prepared? So here for the production of biofertilizer we require skill. So if we are skillful in the production, then it will be easy to handle it, otherwise it will be difficult. And same way here, as it is living fertilizer, as Archana had discussed, it is difficult to store as compared to chemical fertilizer. Now here whenever we'll see here some constraints are there. Now what does it mean? That biofertilizers are not popular because of some difficulties, that some of them are as follows, that inadequate popularity is due to that they cannot show instant and dramatic response. That is same as antibiotic. We are trying to take antibiotic regularly. Why? Because it will suppress our disease very easily. Now instead of that, if we are going for homeopathic or Ayurvedic, okay, as compared to antibiotic, it will remove your problem from baseline, but it will take time. So same way here, whenever we are going to use biofertilizer, it will take time, it will not show instant and dramatic result. Inadequate awareness about its use and benefits. So here people, or mainly farmers, they are not much aware about the use of that. Now same way, lack of promotion, extension and insufficient publicity. Now if we want to improve use of biofertilizers, we have to try to create such type of program by which we can improve awareness about the biofertilizer. Same way, lack of availability of quality products in time, because as it is living organisms, if it will not remain functional, then it is of no use. So it should reach in active form to the farmers, then only it will remain active. So by that I'll conclude my topic, that try to use biofertilizers for healthy and nutritive soil. So now I'll hand over it to Archana.
Okay, thank you. So what we have discussed in this discussion of biofertilizer, starting from the introduction of biofertilizer, live manure is there, live nutritive elements are added in such a way to the plant, the organisms are helping to leach out different elements, different chemical that as such not available by the plant from the soil only. They are present in the soil, but soil is there having the particular nutrient, and root is there, plant root is there, but root is not able to take up that nutrient. So microorganisms are helping the root to take up that nutrient, and because of that action the plant growth, the crop production can be enhanced. So different types of the fertilizer discussion is there, how to produce them, how to apply them, all the thing has been discussed. But by this lecture the talk of biofertilizer cannot be completed. Still, when you are not applying the biofertilizers in the routine particular villages that are using different chemical fertilizers, that is very harmful, that is not at all eco-friendly. So chemical fertilizers use should not be there, and instead of the chemical fertilizers, biofertilizers production should be enhanced. And because of that we can say that if you pass on this message to the all, then and then only the biofertilizers use can be there by each and every farmers, and the crop production can be there. So thank you, and give the message to each and every farmer that you should use the biofertilizer. Thank you.
Thank you.